My name is Ruben Munera, and this afternoon I will be talking to you about barley-spent grain as a carbon source in the production of and the sustainable production of single-cell oil using orantokitron limacinum. Right? I work in the bioprocesses research group. This group belongs to Universidad de Antioquia. We are located in the city of Medellin, in the country of Colombia. Okay? So today, I want to tell you that humans need to consume omega-3 fatty acids. And these omega-3 fatty acids are mainly found in fish oil. That's the main source that we have right now. These omega-3 fatty acids are long-chain polyunsaturated fatty acids, and they are made mainly of DHA and EPA. These two fatty acids here, they have proof that they have a lot of benefits in the human health. They are linked to brain and retina development in infants in the early life, and later in life they are linked to inflammatory cardiovascular disease prevention in adults. Now the problem is that Western diets don't consume enough of these omega-3 fatty acids. We tend to have diets that are rich in omega-6 fatty acids, but not in omega-3 fatty acids. We don't consume that. And now that I'm here in Sonora, they tell me that you have like a very rich diet in meat. Okay, so not a lot of consumption of fish. So that increases like the gap between the consumption of these two fatty acids. Okay, so we need to find ways to produce that. So, as I was telling, the main source of these oils is fish capture. Okay, and in the last decades, according to the FAO, an institution of the UN, the capture of fish has remained stagnant. Okay, so we are capturing about 10 million tons per year, and this being like the same thing for the last years. And the oil production, the fish oil production, has remained between 1 and 1.25 million tons a year. So we are having the same amount of fatty acids from this fish oil, but we know that population continues to grow and the demand continues to grow. And more importantly, this fish that is being captured, back in 1975, we said that around 7% of the fish stocks that were captured from the sea were considered overfished. This trend has increased, and by 2015, we had around 35% of the fish stock was considered overfished. And if this trend continues, we can see that we are depleting the oceans from this source. So we need to find new ways in which we can have this fish oil. Not the fish oil, but the omega-3 fatty acids that we need to include in our diets. Okay? So in this way, we have seen that there are some microorganisms that they are called marine microorganisms that have a very similar composition of these fatty acids. The trostochithrids, which is a family which we can find schizokitrum and orantokitron limacinum, they have a really high production of DHA. They have between 40 to 50% of their weight of the lipid production is DHA and EPA. And this is even better of what we can find in fish. Several species of fish, they have between 15 and 30% of their lipid content is DHA and EPA. So these sources that we have here, they have shown to be a better source for these kind of essential oils. Now, what is the problem with these? The problem with these trostochithrids in this family of chromists, which is the type of microorganism, is that their production is mainly, their culture uses glucose as their carbon source. And then glucose is a very expensive carbon source, and this makes this type of cultures economically unfeasible. So what we need to do is to find new ways or cheaper ways to produce these fatty acids. So having this in mind, what we wanted to do is to find and to evaluate alternative carbon sources that allow us to produce these single cell oils that are rich in DHA and EPA, and that are they can act as a substitute for glucose. So in this way, we found that orantoketone limacinum is a really good producer of this type of lipids. Okay, and what we did towards this goal was to search around the area of Medellin. We looked for different types of carbon sources. Then we characterized the sources, and finally we had different media that we could use to grow orantoketone limacinum. So among the sources that we studied, Barley-Spent Grain showed that had a promising characteristics as a replacement for glucose. When we did the characterization of the Barley-Spent Grain, we found that it had a cellulose content of 47.36% and heavy cellulose of 42.73%. So that showed a higher potential, or a very good potential, as a glucose source. So what we did after that was we had hydrolyzed this carbon source. We used an enzymatic hydrolysis. We applied these conditions. We ran the hydrolysis for 60 hours at 50 Celsius degrees. And what we got at the end was this medium that was rich in glucose. And after that, we had two media. So we have one media that was our basal glucose medium, in which we had a concentration of glucose of 50 grams per liter. And then we have our alternative carbon source, which are Barley-Spent Grain, and we achieved a concentration of glucose of 20 grams per liter. Now, we also estimated the cost of these two media. And we found out that the glucose, using glucose as a carbon source, cost per liter $7.69, and our medium was a little bit cheaper, was $5 the same liter. Okay, so here we can see a decrease in cost. And then previous studies that we did in a research group had demonstrated that the best conditions for the growth of orantoketone limacinum was a carbon to nitrogen ratio of 25:1, temperature of 23 Celsius degrees, and agitation of 160 revolutions per minute, and a seawater concentration of 50%. This ratio is very important because it has been demonstrated that nitrogen depletion in the medium causes accumulation of lipids in the microorganisms. So we need to have a ratio that allows for the depletion of nitrogen to further accumulate lipids. Okay? So after this, we measured glucose. We use a glucose oxidase kit. We use modified yeast assimilable nitrogen for measuring nitrogen, and we use freeze-dried for determining the biomass in the dry cell weight. So what we had at the end was two kinetic studies. To your left, you can find the glucose kinetic study, and to your right, you can find the Barley-Spent Grain kinetic studies. So after we had this, we started to compare the results that we got. So both medium, we saw that here on the green line shows this is nitrogen consumption. So both of them show that nitrogen depletion started around 72 hours of culture, when the culture had started. And then something very interesting happens with the glucose. This is the biomass accumulation, so we can see that it's growing. But after nitrogen is depleted, we can see an increase in the weight. And we think that this is due to the accumulation of lipids. So after nitrogen is depleted and the microorganism starts to accumulate lipids, we can see it in the increase in the dry cell weight here. For the barley-spent grain, we didn't see this behavior. We saw that it continues to grow at the same level. We don't see this jump. But we still see some nitrogen depletion as we are measuring. So we think that what is going on here is, given that we're using a complex medium, okay, such as an industrial waste, which is very important because we are not only producing lipids, but we are finding solutions to wastes that in this moment are being dumped from the industry, in this case the brewing of beer. So it's complex, and it not only has the nitrogen that we supply to the medium, but they also have some compounds and some nutrients that we didn't account for at the beginning. So it's richer in nutrients that we thought, and therefore we don't see like this height that we can see here, but it continues because it's still, even though the nitrogen we supplied was depleted, it continues to grow. And also, we saw a very interesting behavior. So for the glucose, as I mentioned before, we fed it 50 grams per liter, and we obtained the maximum biomass was 25.5, half of what we fed. And when we see this comparison to barley-spent grain, we fed 20 grams per liter, but we obtained 17 grams per liter of biomass yield. So again, we attribute this to having a richer medium in nutrients and compounds that come from this complex substrate. Okay, so it was very interesting. So at the end, and this is what was interesting to us, we can see total fatty acid accumulation of 36.31% of dry cell weight using glucose, and we saw 19.55% of dry cell weight when using barley-spent grain. Okay, so in this case, this is significantly lower than the amount of lipids that we get from glucose. But when we started to analyze the kinetic parameters, then we found that there is like a balance in the system. So the substrate, the biomass yield, we can see was significantly higher in barley-spent grain with 0.74 compared to 0.43 that we go from glucose. That's what I was showing before in the kinetic graph. But since the lipid accumulation is significantly lower, as I just mentioned, so having a higher biomass yield compensates this lower lipid accumulation. So at the end, the product yield we can see is very similar in both media. We have 0.15 for glucose and almost 15 also, 0.1448 for barley-spent grain. And more interestingly, we can see that the microorganism adapts very well to both media. We can see that it has a growth velocity that is pretty much the same. It's very similar in both. It's actually a little bit higher for the barley-spent grain. So the microorganism has like a really good response to both media, as we can see here. And as I mentioned earlier, using barley-spent grain lowers the price of the culture. Sorry. So from this, we concluded that using hydrolyzed barley-spent grain is a good replacement for glucose, okay, in the production of single-cell oils with orantoketone limacinum. It grows at similar velocities with both media, so the microorganism adapts fairly well to both media, and using barley-spent grain is 35% cheaper than using a glucose as a carbon source. Okay, and also we know that this stage, the fermentation stage in a biotechnological process is very important when determining if it is feasible or not from the economical perspective. So if we decrease the price of the media, we are getting closer to having an economically feasible process. And if we achieve this, then we move forward to having an alternative to fish oil, and in consequence we can lower the pressure on many marine environments. Okay? So thank you very much for your attention.